At the day of the explosion, these people have no clue of the dangers that they are exposed to. You don't really know radiation is there. You're just going to find out it was there later when it starts to really do big damage to your body. And radiation, ionizing radiation, is not only dangerous to tissues, it's also dangerous to anything that is around it.
Almost 9 times the lethal
And
dose.
nobody really knows what's happening. They just had to leave everything behind and go somewhere. Now, this is what happens after you're exposed to the radiation.
Vomiting, nausea, and diarrhea are followed by a latency period.
So here, at first, these are the pilots. They were dropping the sandbags.
It's only later much more fatal symptoms appear, such as
these are the type of burns that you get from increased level of radiation. They just told, go and do it. Somebody had to do it.
Down to the bone.
So you get these burns that will eat the flesh from the outside up down to the bone. You burn.
30 kilometers east of the plant, the forest has been scorched by the radioactive blast from the explosion.
No, seriously. And then you're gonna see what happens to the people that were exposed to the radiation. What is going to happen, as we're going to see, you have many types of DNA damage that can happen in the body. First, you can have DNA damage of somatic cells. That means all the cells in the body but the gamete cells. And you can have damage in the DNA of the gamete cells. Now, if you have DNA damage in somatic cells, you are in trouble. But if you have children, your children are safe. You do not pass somatic cells defects on the DNA down the line. Now, if you have a certain amount of radiation exposure and you hit the gamete cells, then some mutation is going to happen, something is going to be wrong. Now, everybody here took x-rays, right? One time or another, break a bone, or you have to do dental work, you put x-rays. First thing they do, they put a metal or a lead shield, this very heavy coat, that you say, okay, just wear that heavy shield, and then you take a picture. Why? Because it's protecting most of your important somatic cells and the germline cells in order to be not exposed to the X-ray radiation. Because if you get mutation, then you pass it down the line. Now, this is what happened to the children that were born after being exposed to the radiation, and the type of genetic problems that they got. Okay, here's the helicopter. This one, it exploded. Look at the size of that hole. And this is the guy with the sandbag, dropping the sandbags in. Look at the helicopter. It just dropped, it melted. Now, these are the kids that were born after the parents were exposed to the radiation. It's very disturbing. Let's get to the damage. So, mutations. What are the mutations? Any change made on the DNA sequence is called a mutation. Change that wasn't there before. Okay. Now, if you have a change, I'll show here the bad part of the change. Okay, all the problems, or not all the problems, but some of the problems that can happen in case you're exposed to very high levels of radiation. Okay, so we're talking about the bad parts of radiation exposure and genetic change and DNA mutation, but not all mutation is bad. Actually, mutation in the DNA sequence is what causes evolution. So if DNA is always the same, it never changes, the species will never quite evolve. If you have some type of DNA mutation, you actually are developing new alleles for a certain gene, and therefore you're developing variation, diversity. So examples of changes in alleles that lead to evolution or that can be detected would be in the case of the sickle cell anemia, that you have a mutation in DNA that changes the property of the protein. And also we see that the DNA has so many parts that are just never transcribed, never translated, and that acts more or less like a buffer zone, meaning that if you have DNA mutations that are going on in the body and it hits the parts that are never transcribed, never translated, it doesn't matter, because it's just like the mutation never happened. It's the idea of, if you have a tree in a forest and it falls, does it make a noise? Does it matter? Well, if you look at the whole forest, it's probably not going to matter. So just taking a look at the DNA, you get a mutation on a piece that is not important, then you're not going to really have problems with DNA mutations. Now, mutations are permanent. They cannot be reversed, usually, by themselves. And so you have convenient type of mutations. Like, for example, in the lab, when we're studying the antibiotic resistance of certain bacteria, I said, okay, take the antibiotic for 3 days, and then if you stop, you're going to allow some bacteria to grow with the resistance to that antibiotic. But then, did the antibiotic cause the resistance in the bacteria? Well, actually, no. The antibiotic does not cause the resistance to show up, but it will allow for everybody who doesn't have the resistance to die. So if you kill everybody who doesn't have it, you only keep the ones who have it. So that's a type of selection. Now, the book is also describing mutations that can be classified in different ways. Because at first, scientists could not quite look at the sequence of the DNA. So they would look at the chromosomes, and then they'll see something that didn't look right, and they say, oh, look at that, it's a chromosomal mutation. But then after, when they knew there was a problem with some type of gene expression or with some type of protein that was translated not in the right way, they would say, oh, it's a gene mutation. It's in the gene, DNA mutation. But then if they could see it in the microscope, they could look at the chromosomes and see some mutations with the tags or probes, or just the shape of the chromosomes, they'll say, oh, it's a chromosomal mutation. But after all, they're all the same. It's changed the DNA sequence. And now this type of reference doesn't quite make sense, because you can see the mutation, yes, in the microscope, but you can also see, if you do sequencing, and you see the letters, you see the nucleotides that were mutated. So the visualization of it, as they had it in the beginning for this type of classification, is still kept due to a tradition, but it is not just due to, oh, you can see, you cannot see, because now we see the sequence anyway. Now the idea is that chromosomal mutations are very large, as we're going to show, and then gene mutations are small and only in a very defined segment of the DNA.